All right, thank you, thank you. So again, good morning. I am Brandon Henry. I am an assistant professor of biology in the Department of Natural Sciences. I will be serving as one of the presiding officers this morning. To my immediate left, I have Dr. Kizzy Donaldson-Richard, an assistant professor in the Department of Criminal Justice, who will also be serving as one of our presiding officers. It is our pleasure to welcome you to this year's Undergraduate Research Symposium, where we celebrate the creativity, curiosity, and critical thinking that define the spirit of academic inquiry. This year's theme, Research, a Foundation for Innovation and Change, captures the essence of what brings us together today. Research is more than data and discoveries. It's the spark that drives progress, whether in the sciences, humanities, arts, or social sciences. It provides the groundwork for ideas that challenge the status quo, inspire solutions, and shape the future. Today, we recognize the important work of undergraduate scholars who are not just learning about innovation, they're creating innovation. Their research represents bold questions, thoughtful exploration, and a commitment to making a difference, both in their fields and in the world. Thank you for being here to support and celebrate these emerging thinkers and change makers. Let's be inspired by their work and reminded of the power of research to build a better tomorrow. Now, in your program, on the second page, there's the agenda, and we will follow it as printed. We will now have our welcome and acknowledgments from some of our ASU administrators. We have Dr. Rhonda Porter, our interim provost and vice president of academic affairs. We have Dr. Melanie Hatch, the dean of the College of Arts and Sciences, and Dr. Nanika Osakwe, the director for the Center for the Transformational Student Experience. So Dr. Porter, Dr. Hatch, and Dr. Osakwe, please come forward.
Good morning. First of all, thank you for coming to this event. I know that it is early, I know that it is Friday, but I do thank you for spending this time with us. We have been putting on this program for the past 13 years, and it's a wonderful program. We like to highlight our students, we like to highlight our faculty, and we have a wonderful guest speaker. It just so happens that this year our guest speaker is an ASU Golden Ram, so let's give her a hand already. We want to welcome her back to the unsinkable and indestructible Albany State University. Our theme for today is Research, a Foundation for Innovation and Change. And it pleases my heart that our students and our faculty are creating change, making a difference, learning new things. And I was going through the program, and some of the titles of these research presentations are just remarkable. It's some very large words, some very intense science things going on, and I just really enjoyed just looking at the titles and just knowing what our students and our faculty can do. So I want to thank our faculty for giving of their time and giving of their experiences and supporting our students. It is always great when our faculty and our students have a different relationship, not just teacher to student in the classroom, but when you embark upon a research project together, it does so much for your relationship and for your academic experience. So we do support our undergraduate research program, and we want to thank Dr. Osakwe and her group for putting this together. And I just encourage you all to listen and take in what the speaker has to say, ask questions in the presentations and in the poster sessions, just really engage. And of course, be inspired by the remarkable projects that our students have undertaken. Again, thank you for supporting and being a part of our annual Undergraduate Research Symposium. Everybody?
So I'm Melanie Hatch. I'm the Dean of Arts and Sciences here at Albany State. And you know, in the time that I have been dean, I've had the privilege of attending concerts, going to exhibits, attending presentations for capstones. And I have to say that I started thinking, you know what? Arts and Sciences invented experiential learning. But I have to say that deep down, there is something special about conducting research and scholarship. That is one of the cornerstones of academic work. And as I looked over today's agenda, I was highly impressed by what you have all accomplished with your faculty mentors. The skills that you get by conducting research, when you think about really focusing on a question, wanting to find an answer, you're really pushing the boundaries of knowledge in all these disciplines. And the skills that you do obtain by doing research will just be a tremendous help for you, whether your career is in academics or takes you on another path. So again, I'm looking forward to the rest of the day. I really hope that you have enjoyed the work that you have done, that it's made a difference in your life, and that perhaps when time is right, you'll pay it forward and take someone under your wing in the future and maybe do a research project with them. So thank you all for being here, and enjoy the rest of the day.
Good morning, everyone. I am so excited for today, where we are all gathered to see the outcome of over 2 semesters of work from our students. Undergraduate research is a high-impact practice in education. This means that students who undergo undergraduate research actually are retained. They don't get to drop out. They actually get to graduate. They actually get to be the ones to mentor other students. So we are so profoundly appreciative of all the students who are here today to share the outcome of their research. And this is not to say that they are doing this alone. Without the faculty mentors, we will not have them to get to the point where they are today. So, thank you so much, mentors. Thank you so much, the committee members. And thank you to faculty and staff, all the departments who have made today possible. We know that research is a foundation to innovation and change, because there lies the ability to generate new knowledge, validate ideas, solve problems, and systematically support evidence-based decisions are derived from research. And so that is why we all join hands in making this work. Some of the outcomes of research, we see that every day. Even the campus that we are in here today is an outcome of research. The labs that we have are all outcomes of research. Informed decisions we make come from data from research. So research is inevitable. Through research, we are able to identify problems. We are able to engage in thorough investigation of the problems, and we are able to come out with outcomes. And that is why we are so happy that a lot of our students are engaged in research. Through research, we get to validate ideas. We get to know which ideas to throw off the window and which ideas to pursue. Through ideas, we have continuous improvement of standing innovations that we already have. We don't just stop there. We keep researching and improving. So research is inevitable. Through research, we adapt to change. Change, social change, environmental change, economic change. It is research that makes this possible. So without doubt, research is a foundation to innovation and change. And we appreciate all our students who are engaged in research. The 13th Undergraduate Research Symposium did not just start today. It started 13 years ago. And the students who are engaged in this cohort started their work last fall. They had to submit their topics. Their topics had to be verified through their mentors, and they had to present proposals. These proposals had to be verified and accepted before they were invited to engage in the research. So they did this for a whole semester with their mentors, the faculty who dedicated their time to see them through. And so this semester, they finalized their research. And today, we have them ready to share their presentation in two modes. They are going to do oral presentation, where they share their PowerPoint, share their main topic, what's the rationale for it, what's the method they used, what's the finding, what's the implication of their findings. They will share that, and based on that, the reviewers and judges will select the best, the second position, and the third position for the two categories we have. We have altogether 3 cohorts. We have the main cohorts that are funded through the Center for Transformational Student Experiences. These cohorts are the main cohorts. But we also have cohorts coming from grants from faculty, PIs. So we also have them. If you look at the program, towards the end, you see the grants that are listed and students who are working on that. We also have research from global learning. Those faculty members who were engaged in internationalizing courses, which means including global learning, intercultural, international learning into courses, because not all our students are able to study abroad, but our faculty have found innovative ways using research and various pedagogy to get students to learn about other parts of the world, economic situations, historical situations, environmental situations happening in different parts of the world. They get to do comparative studies, and we've had these types of research done over the past years. Last year, we had Dr. Jones mentor over 20 students through these cohorts. This year, we have Dr. Owusu Ansah mentoring several students in internationalizing courses through research based on different countries. So all these are what we are going to be observing today. So altogether, we have 52 students who are going to be sharing their oral presentation through PowerPoint. We have those who are going to be doing the poster presentation. That will be in Fine Arts Building across the hall. And because of the renovation going on, we couldn't use the space here. But over the other side, Fine Arts Building, we have the poster session going on. And we implore all of you to please stay till the end. After the opening ceremony, we will have a short break, and then after that, we'll start the oral presentation immediately. The oral presentation is going to take place in 5 locations. We have 141 Billy C. Black. We have 143 on this side. We have this auditorium, and then we have 205 and 203 of Fine Arts Building. So these are the places where the presentations will take place. So I want to use this opportunity to say a big thank you to my staff at the Center for Transformational Student Experiences. Without them, this event will not be possible. And do I have Dr. Milesh, Ms. Reed, and Israel? Can you please stand up for them to please acknowledge you? And please, a round of applause. Okay, and Israel is our graduate assistant. I want to also use this opportunity to say a big thank you to the Undergraduate Research Committee members. If you are here, could you please stand to be acknowledged, committee members? Keith is here. Thank you all so much for helping us to come up with decision and come up with this program that we have right here. So without much ado, I want to very, very much acknowledge the presence of our distinguished guest speaker, Dr. Melissa Davis, who is the director for the Institute of Transformational Genomic Medicine at Morehouse School of Medicine. She is an alumni of ASU, and we are so honored to have her accept and be here today to be part of this audience to give us an exceptional keynote address that we've been looking forward to. Please, could you please give her a round of applause? And she is here with her 2 kids. Can you please stand up too? Thank you so much for coming to support your mom.
Oh, wow, thank you.
Okay. Thank you so much.
Yes.
They are going to read your bio before you come up.
Okay, okay.
Thank you. Okay, so thank you all so much.
Great morning, everyone. I'm going to introduce the keynote speaker, Dr. Melissa B. Davis. Dr. Davis is the director of TGM at Morehouse School of Medicine. Team lead and center grand challenger team, and distinguished investigator with the Georgia Research Alliance. She also served as the scientific director of ICBSC, the director of Health of Precision Medicine, and the associate professor of Cells and Development in Biology in the Department of Surgery in New York. For several years, she has been joining the MSM. While in New York, she served as the Cancer Genetic Scholar co-leading Project Center. She currently holds an associate faculty appointment. Dr. Davis received her PhD in molecular genetics in University System of Georgia in Athens. Now I welcome Ms. Melissa B. Davis.
All right, good morning, guys. I am really excited to be here. I give a lot of talks, hundreds, I feel like, every year. Trying not to travel as much so my kids can see me more. But I'm really excited about being here because I used to be right where you are. So when you look at my bio right now and you see these words and titles, each one of them represents a story, a challenge, I'm even gonna say, throughout my journey. So I'm gonna talk to you today about the research that we're doing right now, but I'm gonna put it in the context of what my journey has been. So when I was here at Albany State, let me start there, I'm from Albany, Georgia, right down the road at Riverdale, that's where I grew up. I went to Westover. Got some Patriots in the audience, maybe? Yeah, all right. When I first came to Albany State, I was pre-law, because you won't know this, but there was a show on TV called LA Law, and I wanted to dress nice and, be a lawyer, and that's all I really knew. Then I realized that it was boring to me, no offense to the history department here, but, I would fall asleep in the classes, and I was really a scientist at heart because my mother was a science teacher. And so I spent countless hours in the back of her classroom just pillaging in her little storage closet, setting up little experiments. She let me do whatever I wanted to do. And I started to feel some sense of responsibility while I was at Albany State. Responsibility to my community and my family. And I felt like whatever I'm gonna do for the rest of my life, I need to make sure that it's gonna be a contribution to lifting our community higher, because there were a couple of, let's say, political events that occurred while I was at Albany State. So was the flood of 1994, by the way. And I decided that I wanted to be a clinician. Now, that was really my only sort of perspective into science. Either I was gonna be a nurse or a doctor. I really didn't understand what being a scientist was about until one of my professors was so thoughtful about the learning experience of students that he put in the hard work of getting a grant for us to be able to have a stipend and research support to go do summer programs through a mechanism called the Minority Access for Research Careers. So then I was a MARC scholar. And even the fact that that program exists means that there were people in Washington who were lobbying for minority scholars to have access to research careers. And I want you to understand that in the context of the opportunities that you have, because in order for you to do your research, that means that there's a professor somewhere who is taking their time to write 50 pages of a grant proposal and going through a lot of ridicule and critique and having to, resubmit it. And it's a lot of hours, it's a lot of hard work just so there's enough funding for you to be able to do what you do. So cut to the chase. I decided to do research. Now, originally I joined the MARC program because they gave a stipend and I wanted a car. And because of the stipend, I was able to buy a car, and I didn't have to have a side job, just to pay for my shoes. I had a shoe problem at the time. And, but there was a requirement for me to go away and do a summer research opportunity at one of the major institutions, and the institution I went to was Ohio State, the Ohio State. So when I got to Ohio State, and again, I'm from Albany, and half these buildings that you see didn't exist here. And so I was used to a small college in a small town. When I got to Columbus, Ohio, and there was over 100,000 students, including professional students. And the classrooms were double the size of this, right? Every class that I entered, it was an auditorium style. So there were hundreds of students in each class. It blew my mind, but what actually changed my perspective of my career was being in the lab and being in a building filled with laboratories and understanding that all of the thousands of people in this building are all doing some kind of research. And then everything started to click for me. So all of the information that's in your textbooks are the results of experiments and the research that PhDs did in labs like the one at Ohio State, but also the result of PhDs in small colleges like those here at Albany State, at Ohio State, but also here at Albany State. So what I learned was I studied spinal cord regeneration in salamanders. A little bit of trauma there because I, didn't want to touch them, but I had to actually do surgical procedures on them. So that was nice. And I remember the moment that I thought, I want to be a scientist. After we did these, it took me weeks to get to the point of having the tissue on the slide and seeing illuminated the expression pattern of this gene that my PI, Dr. Roy Tassova, was studying. And he said, in this moment, Melissa, you and I are the only two people in the world who know that fibronectin 11A is upregulated during spinal cord regeneration, and it could be the key to solving paralysis in humans. And I was like, oh, wow, so we're going to get a Nobel Prize? He was like, no, not quite. This is just one step, this is one experiment, but I'm just saying, this will be a paper, you'll be an author, and who knows what that could mean for the rest of your life. And that feeling that I had in that moment of knowing that this gene could be the key to solving spinal cord paralysis changed my life. And so you'll have those moments, and those are the moments that drive a scientist to move forward, because you'll fail. And you'll take months and months trying to get to that one moment where you discovered something, but it's worth it. Now, the talk that I'll give you today is called The Grand Challenge of Unraveling Social versus Biological Drivers of Racial Disparities in Cancer Outcome. Every one of those words is intentional. What I want you to appreciate, I have some animation in a few, so it may be a bit difficult for you. I apologize ahead of time. Okay, so I want to talk to you about breaking barriers, because after I finished my PhD at UGA, I went on to Yale School of Medicine. And then from there, I went to Chicago. I went to the University of Chicago. From there, my first faculty appointment was back at Athens at UGA. From there, I went to Henry Ford Health Systems. From Henry Ford, I went to Weill Cornell Medical School in New York. And now I'm back home in Georgia at Morehouse School of Medicine. And this long journey through Ivy League institutions, private institutions, back to my state institution, and now back to an HBCU, every step was intentional and almost forced. Okay, there was a barrier at each one of those institutions that I had to overcome or navigate. And praise the Lord, every time I got over a barrier, it landed me in a higher position. But I was only able to get to a higher position because I had, support. I had people around me who understood my mission. They understood and agreed with what I wanted to study, even though there were others who did not necessarily agree. So let's think about healthcare in the United States. Okay, so I study racial disparities, and right now that's a trigger word, right? So the president doesn't like anything to do with DEI or health disparities or focusing on underserved communities. So we now have to start rethinking. I'm facing a new barrier. So now we have to start rethinking the way we talk about the work that we're doing, but the mission is still the same. So what I'm showing you on this map, and I'm going to rush through some of this because I don't have time to tell you about all of it, and I want you to ask me questions, okay? So I won't tell you everything, but I want you to ask me questions later. So what this map shows you is county by county the rate of poverty in the United States. Now, I'm overlaying currently where all the African Americans live in the United States. And so what this should show you is there's a clear correlation with socioeconomic status and whether or not you identify with being an African American. That means that there are systemic correlations to marginalizing this particular race group. Now, when we think more about, let's say, healthcare in America, we have to do that in the context of history. And I think that's where our power lies, in our community. And I'm going to have a real talk here. Now, sometimes I don't talk about these specific topics in the context of my research, but I feel like I'm at home, and I want to empower you with some information. So if we think about African Americans in the United States, we know that we are descended from those Africans who were stolen, right? So you're a part of what they call the lost tribe. And I spent a lot of time in Africa. And if you look at the history of the medical treatment of our population and others, okay, we don't have a monopoly on being marginalized. What we see is that from when emancipation occurred to when the syphilis trials occurred was in a single lifetime, okay? That it was only between 30 to 60 years there. And when we look at when the syphilis trials ended, again, that is within a lifetime of this moment that we live in right now. And so oftentimes when we hear about the Emancipation Proclamation, the Tuskegee experiments, it seems like it's in our distant past, but we're still touched. We're not even a full generation away from some of those atrocities. And so those are the things that we have to try to overcome. And embedded in the concept of why atrocities were allowed on marginalized populations is this concept of colonialism. It is a process of taking over something that doesn't belong to you and then making those who you are oppressing feel like, well, it's their own fault. And so I put that in the context of cancer research because in the 1980s, the National Cancer Institute took on the responsibility of trying to explain why over a century the Black community seems to be dying more of cancer. If I showed you the statistics of every type of cancer that's surveyed, Black patients have a higher mortality rate in every single category, despite not being the highest incidence. And so that means we get cancer, especially breast cancer, less often, but we die from it more often. So they did this sort of landscape analysis. And I have the title of the slide being Landmarks versus GPS, because I want you to appreciate that when I was driving my car, I had a big atlas in the back of my car, okay? I had to like map out where I was going, and if I had to get directions, people would tell me things like, the oak tree on the corner across from the McDonald's, you're gonna make a right there, right? So you would get landmarks of where you are, but landmarks change over time. But what doesn't change is the geolocation on the Earth's map, right? So if we use GPS, that is a precise measurement of where you are and where you should go. And it doesn't depend on the environment around you. So a lot of what they found in those studies when they had, I think it was 17 different institutions looking at this problem of disparities, and I'm gonna tell you from a telescope because they weren't embedded in the communities, they were studying disparities in a population that they weren't a part of. What they came to find was there were barriers, surprise, to healthcare. They found that there was a delay in the time between a patient was diagnosed and when they got their first treatment. There was even a time of delay of getting the diagnosis from the screening that was allowed. There was a delay in getting access to screening. And they all attributed this to socioeconomic status and the fact that maybe lower education in these populations became a barrier to them having enough money to have access to good health care. But that's not all of the story. So as a scientist here at Albany State, one of the things that I learned in my genetics class was that there were groups of individuals who were studying why people had criminal behavior. And they had come up with this theory that there was a genetic underpinning, especially in Black populations, as to why they had such a sordid criminal history. And I felt that this was improper science in my heart. Now, I didn't know any of the words in the publication. I didn't know how they did the study, but I knew that I had to do something. And one of the things that we seek out is a more in-depth understanding of when there is a superficial analysis of a population. The superficial analysis is something that's at an area level, and you're looking from a distance, and you're seeing things that are correlated, and then you start to blame the population for the life that they live. But if we look more carefully at the entire continuum of cancer care, from prevention to detection to treatment and survivorship, there are embedded in the system barriers that are marginalized systemic barriers to these populations that prevent them from even attaining what they may be seeking. Now, again, I'm gonna rush through some of these, but let's have a discussion about it later. I'm trying to click, I'm sorry. All right, so is it only SES? It's not. So in these two, and I hope you can see it on the screen better than I can. I have my glasses on, but I can't see it myself. But I'm showing you the mortality rate broken up by race for prostate and breast cancer. Breast cancer is at the bottom. Now, what you should see here is that there's a huge gap between the blue and the purple lines. So the blue line represents the African Americans, and they have the higher rate of mortality, in, again, prostate and breast cancer. But in that breast cancer line graph, what you see is that prior to that first line, it didn't matter if you were Black or white. All patients had pretty much the same opportunity to survive a breast cancer diagnosis. What happened at the advent of that sort of divergence where the disparity started was targeted therapies became standard of care. Targeted therapies that were designed to treat breast cancer as we understood it at the time. They were endocrine or hormone-targeted therapies. And so at that time, there was this stark decline in mortality, but only in the white population. For another 2 decades, Black women still died at the same rate. And then we finally got access to that standard of care, and you start to see a decline in the mortality rate in the Black population, but there's still a 30% gap in the mortality. And there's still a large gap in the prostate population. But even within that blue and purple, there's a separation in this graph, and it represents whether the patients lived in poverty or affluence, because African Americans are not a monolith. We are in every strata of the socioeconomic status. We have top earners now. Black people are at the highest level of socioeconomic status. And so if socioeconomic status was really the barrier to healthcare, then we should see a separation of survival. But what this graph tells us is that Black patients who live in affluence and wealth still have a higher rate of mortality than white patients who live in poverty. So that tells us that it's not just about socioeconomic status, that there's something else embedded in this. A statistical analysis, and I'm going to run through this, is done to show you that there are multi-factors that actually lead to the disparities that we see, and some of it is biology, because there are differences in the tumor types of breast cancer. So when you get a breast cancer diagnosis, the first thing they'll do is look to see whether or not you have 3 hormone receptors present on your tumor. And this is because there are treatments for the tumors that do have the receptors. But if you don't have these receptors, then you have triple-negative breast cancer, which happens to be an earlier onset disease, meaning you get it at an earlier age. It's one of the most aggressive forms, meaning it grows really quickly, even between screenings, if you're going through mammography. And there's no targeted therapy for triple-negative breast cancer. Black women get it twice as much as white women. And then I just told you there's already a delay in the detection of it, and it grows really rapidly. One of the first publications that I did was at the University of Chicago, where we showed that the disparities that we saw in breast cancer at the UChicago health systems, where all of the patients were treated by the same doctors, were specifically in the category of patients who had ER negative or estrogen receptor negative breast cancer. That means that the treatments that they gave to the ER negative patients worked pretty well in the white patients, but did not work in the Black patients. That was the first clue that there must be something different about these tumors in these populations. And in fact, when we did another study in New York and Detroit, and this is about 20 years worth of data, what we found was that, again, there's this huge disparity in survival in the Black women who have triple-negative breast cancer. And what these 2 lines represent is whether or not they were early detected or late detected. And so what does this tell me? To see the disparity here, this huge gap of survival only in the Black women, means that the treatments for triple-negative breast cancer don't work as well in this population. But how do we even know this, right? Why would the treatments not work the same? Well, it turns out, and if you never heard this term, I think some of you must have, in pharmacogenetics, we know that our genetic background can sometimes determine whether or not a drug is metabolized the same in our system. Well, the drugs that are used for chemotherapy happen to cause cardiotoxicity in our population more frequently than it does in the Black population. And this is literally associated with our genetic ancestry. So what these bar graphs represent is the frequency of a known gene mutation that creates a sensitivity to a cardiotoxicity sensitivity to systemic chemotherapies. Now, these genes are tested in pediatric patients before they're given chemotherapies, but they're not tested in breast cancer patients before they're given these chemotherapies. So some of the mortality that we see comes from adverse events in the Black women who are taking chemotherapy for triple-negative breast cancer. And this has been shown specifically by another African-American woman who is studying this trend currently at VUMC. Now, the relentless pursuit of knowledge. You have to be relentless in order to achieve the information that I'm talking about today, because you will come across people who don't want you to look that carefully, right? In order for us to have the information that I just showed you, we had to track race, we had to track the outcomes, and we had to be allowed to publish that within a system, a healthcare system, the doctors are treating the patients the same, but the patients don't have the same outcome. And that's bad news. Nobody wants that to be released to the general public. And there's always going to be opposition to us understanding what's causing our deaths. Now, The genetic studies that help us understand, for instance, BRCA mutations and genetic risk were pivotal. I've met Marie Claire King. She's one of the leaders who identified the BRCA gene. But the type of information that she used in order to find those genes doesn't exist in our population, because she had to use these very large pedigrees that went back many, many generations, and they had to be well annotated with health history information. And we weren't even allowed to get cancer diagnoses, let's say 4 generations ago. And so we would never be able to detect whether or not there was a familial predisposition in the Black population that was different from the white population. But GWAS studies, or genome-wide association studies, should have helped us overcome that, because it didn't matter who you were and where you are. In fact, we didn't want people in GWAS studies to be related, because the power in a genome-wide association study is having large numbers of people who have different variations in their genome, allowing us to see what correlates with the disease, right? Well, unfortunately, the majority of that data comes specifically from white patients, white participants, the European ancestry background. So these bar graphs that you see to the right represent the fact that European data is 80% of our understanding right now in our knowledge base. That means less than 8% of the data represents 80% of the world. That means we have not captured the genetic background of the world. But all of the information that we have about clinical tools, about genetic testing, is based on the European ancestry, the European genetic background. This means then when we have mutation test panels, so that's the other bar graph here, there's a higher likelihood that the test isn't going to reveal any useful information. We call those variants of unknown significance. So if you have non-European ancestry, and again, we don't have a monopoly on this marginalization. If you're East Asian, South Asian, Latin American, if you're not white, then you're likely to find a mutation in a cancer gene that we've never seen before. And if it's of unknown significance, it also means it's unactionable, so then you don't get to do anything. If you have a mutation in a cancer gene, the arm of the healthcare system that allows better surveillance, more monitoring of your health, is supposed to open up for you so we can catch the cancer early. But it's unknown, so it's unactionable, so you don't get that extra surveillance. But how do we even know this information? Again, it's because we're tracking with labels, data, populations, and now we're being told that we have to stop using those labels. We can no longer talk about men and women in data. We can no longer talk about race and self-identity in data, because they say that's discriminatory. But it's actually revealing. It's a tool that allows us to understand how we can identify barriers. And so this is one of the things I did. Again, a blessing. I was trying to get over a barrier, and in climbing over the barrier, I met Lisa Newman. She was at Weill Cornell. She's a breast surgeon, and she had a global operation of training clinicians of how to treat breast cancer in Africa. She came up with this term she called oncologic anthropology, because one of the things she observed in her training of general surgeons was that when she was in West Africa and Ghana, there was a higher rate of triple-negative breast cancer than there was in East Africa. And when I joined her team, we then measured genetic ancestry. And what we found was that in every country where we could separate the population by genetic ancestry, it was the Black or African component of the country that had the highest rate of triple-negative breast cancer. And globally, the highest rate is in sub-Saharan Africa. When we measured ancestry, we found that West African ancestry was associated with having a higher likelihood of having triple-negative breast cancer. So what does that mean? There's something in the West African genetic background that gives a specific predisposition to triple-negative breast cancer. And this was the first time that genetic ancestry had been linked to a certain type of tumor. Now, what does that even mean, Dr. Davis? What do you mean that if I'm of West African ancestry, I have a higher likelihood? Why is that? Well, in admixed populations like African Americans, your genetic ancestry is spread all over your chromosomes. So what having more of a certain ancestry really means is that you have a higher likelihood of having this one or maybe two positions in your chromosomes that are from Africa, and it's those different alleles or gene types that are giving rise to this type of cancer in a certain context. And so we use genetic ancestry as a tool to help us sort of tease out where in the genome, where in your chromosomes are these genes that are leading to triple negative breast cancer. Now, that concept was, again, challenged, right? I couldn't even get the papers published when we first found this. And so I had to go back to genetics 101 and say, well, you know, there are other gene mutations that are not associated with being African, because that seemed to be the tension in the room, like your blood type. If I look across the globe, I can find that certain blood types are more prevalent in certain geographic regions that are also associated with background from your genetic lineage. And if blood type has a clinical, meaning and is important, then how many other genes or other variants could also have these clinical meanings? And the fact that we can track genetically where these populations came from because of the transatlantic slave trade, that gives us the statistical models to be able to measure in the genome specifically how and where we can track the positionality of a certain risk. So the next step requires a bold vision. I've been told all of my scientific life that I'm ambitious, right? I don't know if it's ambition. I just see that there's a problem that we need to fix. But when you read my proposals, it's like, oh, wow, this is too much, and you're going to see what I mean by that. But that's sort of been the critique that I've gotten, that it's too big. I remember the very first grant I wrote in my first faculty appointment, that's what all the reviewers said. It's too much for this junior person to do. Not that it was wrong, not that it wasn't scientifically sound, it was just too big for her. But God. All right, so meet Standby. This is a team effort of 14 institutions across the world. This is the pinnacle of my success. Now, I've gotten small grants along the way, but this grant is co-funded by CR UK, so Cancer Research in the United Kingdom, and the National Institutes of Health. And it's a $25 million grant that I get to run from a historically Black college. And that's like pinching myself. I can't believe it. The logo that we chose is a lens, and each portion of the aperture represents a discipline that we're using to address cancer inequities at a global level. I'm going to skip this in the interest of time. Here is my team, and you can tell they're very diverse, and they're from, again, different countries all over the world. I won't have time to tell you about all of them, but we focus on the types of cancers that we know have the highest mortality rate in the African diaspora. So that lens is focused on Africa. And with intention, we have this integrated team that works on social determinants of health, exposures in the environment and environmental health, genomics and genetic background and epigenomics, and then tumor biology focused on immunological factors in the microenvironment. Our goal is to study 40,000 patients. 27,000 of them will have cancer, and the rest of them won't, because we need to have a control for the comparison. And what we hope to deliver to the scientific community is a wealth of data that's focused on a population that has comprehensive characterization of every layer of each one of those teams that I just described. And I'm going to walk through for you what each team is going to study. Now, the social determinants of health team is the foundational layer, because we already understand, yes, socioeconomic status does, of course, have an impact on your health, but more than that, there are aspects to social determinants that are actually societal determinants, right? So Lauren McCullough, who's at Emory, has discovered that Black women in Atlanta at the highest level of socioeconomic status are 126% more likely to die from breast cancer than white women. That's the top earners are 126% more likely to die, even though they have health care, private insurance, they're in screening programs. This is because, again, there's probably something underlying the diagnosis that makes it difficult for them to have an effective treatment response. So what they're going to do is reframe the way we even measure social determinants in the Black population, right? There are factors in our population that aren't a part of the equations that are currently used to define terms and indices like neighborhood deprivation. So take a moment and think about, what does neighborhood deprivation mean? The way it's currently defined is talking about how much money you make, whether there's a grocery store near you, if you walk or if you own a car, the school systems that are around you. And so if you take a Black woman out of those characteristics, so, you know, I live in a good neighborhood. I live in a neighborhood where most of the people in my neighborhood have advanced degrees. There's a grocery store less than a mile from me, but I don't necessarily feel welcome in my neighborhood because the majority of the people in my neighborhood are not Black. There's a social isolation here. There's a tenseness, if you will, where I don't feel safe in my environment. And when I go to work in my high-paying job, I'm probably the only one like me in that corporate office. And so I have microaggressions that I deal with every single day. I'm vigilant about protecting my children because of the environment that they have to traverse in these good schools that don't necessarily see them as belonging there. And so all of that then impacts my biophysical. It impacts my stress, my psychosocial stress. But that's just me as a Black woman in America. We have a global study. So Black women in Kenya, Black women in Ethiopia, Black women in Ghana have to deal with genocide. They have to deal with kidnappings. They have to deal with natural disasters where they have no recourse. And so we'll be looking at how the lived experiences of Black women with breast cancer, the lived experiences of Black men with prostate cancer play a role in their survivorship. It's gonna be phenomenal. I mean, these are factors that I wish when Ketanji was getting her congressional hearing, we could have taken a trace of her blood before and after, because I'm absolutely sure her genome was impacted by the stress that she felt during that onslaught that she had from that senatorial hearing. And so then we step out of the societal aspect of how we live, and then we start thinking about, well, what's around us? There are issues in every one of the cities that exist in the United States that still exist today because of historic redlining. Raise your hand if you know what redlining is. Have you heard this term? Great, so I don't have to explain it. Sometimes I do. And so because of the city planning around redlining, we also know that if you live in one of those red areas, because those were the only places you were allowed to live if you identified as a certain race, you're also going to find toxic chemicals in your water. There's more pollution in your air. And so those also impact your risk of cancer. And so as we look at, for instance, the genetic underpinnings of patients who have breast cancer, this is data from one of our earlier publications, we see that imprinted on the tumors of women with triple-negative breast cancer are the comorbidities that they hold. Meaning I can see diabetes in the tumor, I can see cardiac dysfunction in the tumor, obesity in the tumor. That is not a part of any clinical trial in defining the biology of the tumor. And so all of the treatments that were based on the populations of women who did not go through this societal experience are not represented in the understanding of whether the treatments work. So as we think about who the patients are, their genetic background, where they live, we can have evidence now that tells us that the tumors are impacted by who you are and where you are. And this is information we need to start using in the way we achieve better treatment. I'm gonna speed up here. So the second team looks at environmental exposures. Specifically, we're going to be looking at, again, where patients live and what we can detect in their bloodstream. We're using mass spectrometry, which is, of course, a chemical detection of molecules, and we'll be looking for molecules that correspond with what the social determinants team found. And this will be the first time this is done at scale at a global level. And then we have the genomics team. Now, this is all really complicated, but what I want you to appreciate is the fact that I just told you that 80% of the data comes from Europe. Well, 80% of our data comes from Africa. So that means we're going to start filling the gap of information. We're going to sequence the entire genomes of all 40,000 people who enroll in our study. And then we're going to start thinking about the way we analyze genetic information. We're going to change the paradigm. So right now, if you sequence a genome, if you have the experience of working with genetic data, you know that you take the DNA sequence and then you align it to a reference, meaning what is supposed to be, let's call it the dictionary, if you will, of our genetic understanding. But that genetic dictionary isn't very diverse. It's based on that European ancestry. And so I'll show you some data in a moment, but we'll be creating new references that are relevant to our population. One of the earliest studies that we did around triple-negative breast cancer allowed us to identify molecular signatures that were unique to African Americans and that had clinical underpinnings. And so we stand on that study where we used ancestry and race to identify differences in gene expression. So to the left, you see us using self-identified race. The blue versus green represents whether the genes are up or down regulated. Clearly, we see differences in whether the genes were up or down regulated by race. But to the right, we removed race and just used genetic ancestry. And so you should be able to see visually that the clarity and the difference in gene expression was much more stark. The contrast was much clearer when we used genetic information to do the statistical analysis. And so that's what we stand on, the fact that the genetic background of individuals does drive differences in gene expression in tumors. And this is something that we should start assessing more clearly. So the Polyethnic 1000 project was started by a Nobel Prize winner who I get to call a mentor, actually. His name is Harold Varmus. He used to run the National Institutes of Health and the National Cancer Institute. He's the reason we have PubMed. He's the reason we have the National Center for Biotechnology and Information. And in one of my landmark papers in one of the highest ranking journals, we got the cover of the issue, and there's a picture of Africa with all these ribbons of DNA. And again, this is a pinnacle. If my mother was alive, I would put a whole sheet of wallpaper in one of these rooms to show her, like, look, Mama, I did it. I arrived, right? And she's just proud that I got a job, so. But I would have loved for her to know that I was able to spark a new way of thinking about how data should be collected and analyzed for cancer. And so now it becomes a part of the standard pathway of how population-level studies are generated. One of the things we discovered in that was, again, a subset of genes and mutation signatures that were related to ancestry, and I'll come back to that. I'm sorry I'm having such a hard time with the clicker. Guys, I want to scoot through this. I don't know where to point. All right. And the last team looks at tumor biology. Now, they're looking at every single cell. And one of the things we've determined is that the differences in the immune cells in tumors, depending on if you're black or white, can be a distinguishing factor of whether or not you survive. Now, typically, the data that was analyzed before, again, majority white patients, would be at the top here, where you see that red line having a lot of immune cells, and if you have a lot of immune cells in your tumor, that means you have a better chance of surviving. This is because it's an immunological response, so your immune system is fighting the tumor. But if you're black, even if you have a high count of immune cells, it does not improve your survival. That means that African Americans completely lose the survival advantage of having a high immune response. And so why is that? Well, my lab started to look more carefully at the individual cells that were in, say, the black patients versus the white patients. And what we found was that the quality or the state of the immune cells are different. So instead of these immune cells, and even though we have way more than the white patients infiltrating in our tumors, the state of these immune cells is what we call immunosuppressive, meaning they're not functioning to fight the tumor, but in some ways are actually feeding the tumor. And then the last team is patient advocacy. So it's led by this rock star, Ricky Fairley. She has a company that she calls Touch Black Breast Cancer Alliance, and they have a podcast. And what they're doing is helping us shift the power dynamic. So instead of like those Tuskegee trials where the doctors just enrolled people without their knowledge, we're actually sending survivors of the cancers out into the community to talk to people about what their concerns are and to let them know that there'd be no harm. But more importantly, we hope to give them information that becomes integrated into our society, that you understand what we're doing, that you agree with what we're doing, and our interpretation of the data that we've collected, and that way the patient voice can help us change policies. Now, before I leave, I want to tell you what we've learned so far. So I have a different grant called The Dark Side of Breast Cancer, and this is because I am a Star Wars fan, but the gene is also called DARK. And that gene has a mutation that arose in sub-Saharan Africa. And that mutation led to a blood group that's called Duffy Null. And if you're of West African descent, and if you're of the slave trade descendants, then you're probably Duffy Null. Now, having that mutation means that you no longer have the protein expressed on your red blood cells, which is why it's a blood group. But it also means that you have an immunity to malaria, because the protein on red blood cells is how the Plasmodium parasite docks to your blood to cause malaria. So what we found was that having that mutation, and again, it's a survival advantage in Africa, but having that mutation in the context of cancer also means that you have a unique signature of immune cells that have a certain chemokine or inflammation signature that's unique to your genetic background. A signature that actually says that immunotherapies should be working exceptionally well in these patients. But at the time we did the study, immunotherapy was still only in clinical trials, and our population was not being enrolled in those trials. So we could have been saving lives with immunotherapies in this population long before they became standard of care. But again, it takes someone looking at these types of differences in the population to be able to study that. There's a whole nother set of understanding. We use single cell data where not only can we look at each individual tumor cell, but all the other cells in the tumor microenvironment. And now we're starting to understand how things outside of your body impact the cells that are inside of your tumors. And again, because we understand that people live in different and distinct areas that have distinct exposures, eventually we'll be able to predict that because you live in this area of that city and you have a specific genetic mutation, we anticipate that your tumors are going to have a specific profile that would indicate we should treat you a certain way in order to help you survive your cancer diagnosis. And that's important information for this marginalized community, but it's also important for everyone, right? It doesn't matter if you're Black or Hispanic or Cuban or Russian or Chinese or South Asian. If I'm telling you that where you live can impact whether your treatment works, then that formula should be important to everyone, and that is the definition of precision medicine. And so what we're doing now and what we're about to write up and publish, and I'll be ending here, is, well, that's a nice theory, Dr. Davis, but what's the data that shows this? Well, here's the data. So if I look in New York right now at the red line communities where our patients who enrolled in our study so far live, and I ask this simple question: if you live in a red-lined area, do you have a distinct mutation signature in your tumor? Is your environment affecting the mutational I guess we could call it the stability of your genome. And some of these mutational signatures, again, and what is a mutational signature? Let me define that really quickly. So a mutational signature is just the accumulation of a certain type of mutation, right? So you can change DNA in many different ways. But what is changing your DNA leaves an imprint. So if it's smoking, it's a certain type of mutational pattern. If it's UV light, it's a different mutational pattern. If it's obesity, it's a different mutational pattern. You understand what I mean? So now we're looking at those mutational patterns and we call them signatures. And the question we're asking is, does your mutational signature have anything to do with where you live? And the answer is yes. Okay? So now we're starting to see that depending on where you live in New York City, the mutational signature that's on your tumor can be very distinct. And again, we're writing this up right now. Okay, and I think this, the very last story, and I'm gonna skip all the way to the end here, is whether or not this other concept that I told you about. So, and this one is really contentious right now. Like they don't like this story. And by who do I mean by they? I mean all of the people who were doing GWAS studies and genomics analyses all these years and ignoring the majority of the world and saying, oh, it doesn't matter. It doesn't matter if you're black or white. We can study in this convenient population, and it's gonna be the same because our genomes are 99.9% the same, or I think it's 99.3% the same. That's the statistic they tell you. But then I ask, well, how do you know when the majority of the data comes from a certain group? That group is 99.9% the same, and maybe if you throw in another 3%, you don't see a lot of difference. But if we start to increase our scope and expand the information, then maybe there are more differences. But here's the kicker. In order to have that statistic, you have to go through that pipeline that I described, where you sequence the data and then you align it to the reference. Well, it turns out that when you take data from black patients and align it to the reference, and data from white patients and align it to the reference, there are groups of the sequence that don't align. We call them unmapped reads. And you can look in the supplemental data of all of these genetic papers and they'll tell you there are unmapped reads. And typically we just push them aside and we look at what did map because, ooh, look at all of this data, right? I mean, it's billions and billions of data, points that tell us something that's useful information. And the fact that these aren't useful yet, we just ignored that. Well, here I come, sweeping the floor. What's this? What are these reads? Maybe they do have information. And so that's what we've been looking at. And so the first thing that you see here, and the colors represent different genetic backgrounds. And I'm sorry that I don't have a pointer that I can actually show you from the graph, but if you look at, there's like this one bar graph that's flat. One bar, right, that's flat. That's the genome reference, okay? That is the percentage of, when we looked at whole genome sequences from a population of African and African American and Afro-Caribbean patients, that's how much of these particular unmapped reads map to the human reference. But then when we took those same fragments and mapped them to other genomes of distinct African and Native American and Asian ancestry, we saw that they actually mapped. That means that we do have functional information in that data. In fact, I can tell you that if we take all of those reads and we map them first to a set of people of African ancestry, and then a set of people of European ancestry, and then a set of people with Native ancestry, and then to this new reference genome they call telomere to telomere. I don't have time to explain it, but I'll just say that the reference we use had holes in it, and this new reference doesn't have holes. What we start to see is something amazing, which is that in this Venn diagram, all the things that overlap, so what's in the center, that's all of the shared information that no matter what your genetic ancestry is, we all have those regions present. But the things that are outside in those bubbles by themselves, those are things that are unique to that ancestry group. So that means there are unique portions of the genome that are distinct in people of African ancestry and of European ancestry and of Native ancestry. And right now, none of those are a part of any genetic study because they've simply not mapped to the reference that we use. And so we're gonna change the game a little bit, right? We're gonna go back and we're gonna tell the world, well, what's in these bubbles? And we've already started that process. So we know, for instance, that there are transcribed regions. So who's had genetics? What does transcribed regions mean? It means that there are genes that are being expressed. So there are expressed genes in these regions that we've never aligned to the human reference. What are they? What do they do? That could be the key to curing cancer. And we've never even measured the amount of them in any study. But what else is in these regions? We found what we call CPG islands, and that means it's just a track of C's and G's. And structurally, this represents where the start switch of a gene occurs. And there are over 1,000 of them in these unmapped reads. So there are over 1,000 new gene starts that we've never sequenced before. And if we weren't paying attention, if we weren't looking for genetic ancestry as a demarcation of populations, if we weren't sequencing the whole genome of an inclusive and a diverse population, we would have never found them. And so that means all of this sort of antithetical targeting of diversity and inclusion and equity that are being targeted now, they're trying to shut this down, and we can't allow that. And I don't want to get political, but I do want to spark a purpose in these young scientists that are coming behind me, because I'm ready to pass the baton, and I want to make sure that what we're setting up as a foundation of red flags of what's missing in the world, that you pick this up and that you feel the purpose, the sense of responsibility that I felt when I was at Albany State. And together, we're going to get there. And I'll tell you this, they may tell you that you need to stand alone. They'll tell you that in order to be successful, to prove that you are a good scientist, you have to stand alone. You have to be independent, right? This is what I was told. If you collaborate too much, then you're not going to get the credit. We got to get over that sort of colonial mindset. We have to work together. Our culture tells us that if we want to go far, we go together, right? And so there is a science to working in a team, because I can't hold all the talent and all the knowledge. I don't know how to work a mass spectrometer, but I know I need mass spec information. I'm not an epidemiologist, but I know I need people who understand how to design a study that allows you to get power out of the numbers that you're enrolling. I'm a genomicist. I can do the genetic stuff, right? But I need an immunologist to tell me when we have discovered a brand new immune cell type. Like, I'm not sure what I'm looking at, but an immunologist will look and say, oh my gosh, this is a new lineage of cells that I wouldn't be able to describe. And so that's the science of working together as a team, and that's the dream that I had, and I finally got a chance to do it, and then a different president stepped in, but that's okay. If I have to move to London in order to get it done, we will get it done. And so I'll stop here and acknowledge my team from over the years and the many funders that we've had, and our donors, and most importantly, the patients and their families who have enrolled in these studies to allow us to do the data analysis that we've done. So I'm sure I'm over time, but I hope you guys will let me answer some questions. Thank you.